Okay, thank you very much for the introduction. So this has been quite a big collaborative project for myself. Performed over a number of labs, both in France, the University of Aberdeen, and at the Roslin Institute. There's been a number of groups involved in all of this work. Fuck. Okay. Yeah, I'll yeah.
That's also used as a cursor.
Yeah. Sorry.
I'll try and get a new battery for that.
Okay. So, firstly, I'd like to thank Joseph and Ada for giving me an extra 10 minutes because I've actually been involved in 2 work packages in the AQUA-FAANG project. So, my talk's split into 2, and if it's okay with the chair, I'd like to take a couple of minutes after I finish WP3 to just answer a question or two, but it's still fresh in everyone's memory, and then I'll move on to WP4, which was the single cell work we did. So in WP3, the genomic basis of immune phenotypes, I'm going to present the publication that we published just this year in Genomics, which again was this big collaboration project on the conserved, on the conserved and divergent arms of the antiviral response in the duplicated genomes of salmonid fishes. Following on from that, I'm going to talk about the WP4, which was almost solely done by INRA, which was this novel approach to reveal disease resistance. Aberdeen also had a small part in this to do with the gene editing, which Pierre will touch on tomorrow. And all of WP4 is centered around these rainbow trout clonal lines at INRA that have contrasting resistance to bacterial and viral disease. And then within that, I'm going to talk about the single cell data we have from these fish. So just to give everyone, I'm sure everyone knows, but just to explain what ISGs actually are. Whenever we get a viral infection, viral sensors in the cell, such as your toll-like receptors or Rig-like receptors, pick this up and initiate a signaling cascade. This leads to the release of interferons, which can then bind to the interferon receptors. This then leads to signaling through the JAK-STAT pathway and leads to the formation of this ISGF3, or interferon-stimulated gene factor, which is a combination of STAT1 and 2 and IRF9. Following on from the formation of this, it's able to translocate to the nucleus of the cell and then bind to the interferon-stimulated response element of many different ISGs and then promote this antiviral status within the cell and the transcription of hundreds of different genes promoting this further antiviral state. Our aims for WP3 was to compare the antiviral pathways in Atlantic salmon and rainbow trout with a focus on the type 1 interferon responses, hence we used poly-IC. And within this, we had another couple of sub-aims, which was to generate this comprehensive ISG database for salmonids, and then within this, also define the evolutionary relationships of these ISGs, both inferring parology and orthology relationships in respect to the whole genome duplication events. So it's obviously, we've been over what the whole genome duplication events have been, but as many of us can appreciate, the actual complexity in working with these fish that have undergone this 4th round of whole genome duplication. And what I just wanted to highlight as well, much of the work done in the past might not have always focused on the paralogs, but now we really tend to look at what's actually going on between these duplicated genomes. The experimental design of WP3 was the same for every other fish species, but we had the in vivo and the in vitro components of this. So the experimental design fairly simple. We took 6 fish, injected them with PBS, another 6 fish with poly-IC. These were all kept in the same tank for 24 hours. And then we sampled the head kidney, which was split into 3 parts for your RNA-seq, ATAC, and ChIP-seq. We then did the triazole extraction in this and sent it for sequencing. On the other hand, with the in vitro stuff, we had the 6 naive fish, and then this was put into these 6-well plates into doing a control, which was left on its own, poly-IC stimulated. It was 2 million cells per well, and then we did the RNA extractions, the ATAC, and the ChIP preparations. And the whole idea being with it, we have this identical experimental design and downstream analysis, which was done between, at INRA we did the rainbow trout, and at Aberdeen they worked in the salmon. And then following on for this, we did the NF core, the use of these same packages to ensure the same analysis was done, the same mapping techniques, with the only difference being the species of fish. And this obviously mapped onto the latest ensemble genomes, and then using DSeq2 for the differential gene expression. So just an overview of the results. We have the standard criteria of any kind of RNA-seq experiment, p-adjusted of 0.05, and then a fold change either representing a halving of the gene's expression or a doubling. The PCA plots, what they actually showed was we've seen a very clear separation of the 4 experimental groups. So your PBS vivo, vitro, and poly-IC, vivo and vitro, with rainbow trout on the left and Atlantic salmon on the right. The rainbow trout probably a little bit more tightly clustered, a little bit more variation going on in the salmon, but they're still clearly separated. Below, we have the Venn diagrams of the number of differentially regulated genes. What we did see, just in general, was more differentially expressed genes in vivo in both species, but there was more differentially expressed genes in trout compared to salmon. For the actual main bulk of the work, we wanted this focused analysis on these ISGs. So we originally started with a paper from Jean-Pierre Levrault and Pierre Boudinot, which was looking at orthologous genes in zebrafish, ISGs in zebrafish, which had a human ISG ortholog. And this forms the basis of our set 1. So we had 77 ISG families that had orthologs in both human and zebrafish, and then we found the orthologous genes for these in the trout and the salmon. And this equated to 325 orthologs and 348 salmon orthologs. However, the paper they did had used zebrafish larvae, and it also used a couple of different stimulants. They had recombinant interferon and chikungunya virus, which isn't obviously the same as poly-IC, so there's obviously some genes we might have missed. Within this, we then formed a further 2 sets to try and encompass everything. A set 2 was the ISGs in human, which then had a salmonid ortholog, and then a set 3 where we had salmonid or fish genes where we had no human ortholog, and this equated to around 900 genes in both species. So the focus on set 1, which formed really a lot of the bulk of our work, and actually we made these manually created lists of these huge Excel tables which denote all of the orthology, and it combines both phylogenetic relationships and functional RNA-seq data. Within this is then separated into functional categories, so it's a bit hard to see, but I'm going to do a closer up in a minute, where we have secreted factors, enzymes, transcription factors, and any other kind of functional information tied in. It's obviously a bit of a nightmare to look at an Excel table this size that's still even cut off on a slide. So to break it down, I picked a couple of examples to show exactly what we did. So on the very far left, we have the gene family, the ISG gene family. So this could be any of the ones we've identified. The second column then is the gene configuration. So this is a phylogenetic relationship, going from human to teleos to salmonid. So in the classic evolutionary pattern, you would maybe expect we have 1 human gene, 2 teleos genes, and 4 salmonid. We also have the 1-1-2, we have 1 human, 1 teleos, and then we see the salmonid duplication, and then the last category being 1-1-1. Within this, we then have the human ortholog, the ID for ensemble, complete with the gene name. We then have the zebrafish ID, gene name, and we also have northern pike because it's a bit closer relative to the salmon, but still was before the salmonid-specific duplication, and this really helped frame where these teleos-specific and salmonid-specific whole genome duplication events occurred. Within this, we then kind of do a bit of naming on which duplication event these genes might belong to between the teleost and then to the salmonid-specific duplication. And we use a little bar in between some of them in our table to kind of separate out what zebrafish gene and pike gene belongs to which teleos-specific whole genome duplication. Coming on to the functional data side of things, so we have the rainbow trout gene ID and then the matched salmonid, Atlantic salmon gene ID, and then the phylogenetic relationship between these. Within this, we then have the functional fold change data from the DSeq2. Anything not coloured in is not significant. Whereas we've coloured in the significant ones in a range of different values. So yellow being a low fold change, blue being a halving of the gene's expression, orange being a kind of medium fold change, 5 to 20, and anything over 20 fold change we did in red. So it really is quite easy to quickly look at these tables and see what is similar between them and what is divergent between the 2 species. Coupled on with that, we also tried to do a bit of normalized expression to kind of give people an idea of the constitutive level that these genes exist at, which was done by taking the total counts and then also looking at the ELF counts. It's not perfect, but it's a bit of a proxy to give people an idea. One last thing to also note, sometimes one of the species would lose a gene, and this was assessed basically by looking at Ensemble's gene trees and some Scorpio-corrected gene synteny data on whether or not other salmonids had this gene. And if it doesn't have this, then it seems to be an extra gene copy that's appeared. One last category, which I didn't mention, was other patterns, which actually most of the genes kind of fall into. And this is where the phylogenetic relationship was way too complicated to actually try and extract any phylogenetic information. So CXCL11 being an example of this. One human gene, we have 6 zebrafish genes, 3 pike genes, and then a whole multitude of various numbers of salmonid genes. We also tried to do a bit of fuller analysis to try and correlate some of this work. So what we have here is some of the fold change of the significant ISGs in both rainbow trout on the left and the Atlantic salmon on the right. Genes down the bottom represent where there wasn't any expression in one of the conditions, and the same up here on the y-axis. So within this, there was a couple of examples where we highlighted where they were very different, and we do go into much more explanation within the paper of where they were. There was a difference between the two species. So rainbow trout had quite high expression of our ISG12, which was completely absent in Atlantic salmon. And then on the other side, we had other examples as Hills E2 being much less modulated in Atlantic salmon compared to our rainbow trout. So just to give a conclusion to this, we thought this was the first comprehensive study of the salmonid ISG repertoire, both in vivo and in vitro, with these species examined under identical conditions, using this defined orthology and parology to prepare this kind of useful resource for anyone working in salmonid antiviral responses. So I would like to just take a minute there if anyone has any questions about this data before I move on to the second part of my talk, if that's all right.
Okay, yeah, so if there are questions.
I'm just looking at, I'm curious about the orthology relationships of ISGs relative to the average or frequency of different orthology relationships. Do you find you have more one-to-one orthologs or many-to-many orthologs compared to the rest of the genome?
So this was actually a question we did want to answer, but it was a bit too difficult and it was a bit out of the scope. So we did proportions of the different configurations, as we called them. So we were very, very strict in 1-to-1, 1-to-2, and 1-to-4. And actually most of the ISGs all kind of fall into this other category. And it seemed to be a lot of either local duplications or even local gene losses or whatever else.
Because that would have been my guess, because those genes actually have the responsive genes are typically probably have a relatively simple regulation, right? And they probably have only one like cis regulatory model next to them, and they are much easier to actually tandemly duplicate than developmentally regulated genes. Yeah. So I would actually, since you have cage data for some of them, I would look at their promoter types. There would be definitely a huge bias in promoter architectures in this compared to the rest of the genome.
So part of this has already been done actually by the other PhD in the project. He's been looking at some of this as well. I haven't personally looked at it at the moment. But your point is interesting as well, though, because when you said about looking at compared to the rest of the genome, it's kind of, we had the hypothesis, are these genes more selected for because they're obviously innate immunity and maybe more kind of essential in a way. But it's something I think we would have liked to have done, but it was a bit complicated.
So again, I think those genes probably, it's easier for them than average to be tandemly duplicated. And because they are not really, their activity is not really sensitive to dosage very much, and also basically probably of the regulatory configuration, you can cleanly copy them together with practically all the regulatory elements because they are few and close to them probably.
Yeah, it's definitely one part of the story. Okay, I'm going to move on. If there's any other questions, we can come back to it at the end. Is there one more?
Huh?
No, okay, sorry.
Okay. So yeah, I suppose we could go on.
So the second part of the talk was all about these novel approaches, which is all focused on the single-cell data that we did, which is a single-cell RNA-seq of rainbow trout head kidney leukocytes with this contrasted resistance to bacterial and viral disease. And up here, we just have the 10x diagram of how they actually do the single-cell sequencing. So, you know, we have the little barcoded gel beads that come in from the right, and then we have the cells coming up here along with the enzymes. These are encompassed in an oil emulsion, and then there's barcoded within here. These are then reverse transcripted within these little oil emulsions, and then the oil emulsions eventually removed, and we had all this barcoded cDNA for each of the cells. But I was going to be a bit more basic in actually why do we use single-cell sequencing. Now, it's not very new anymore, but I think it's always a bit of a nice introduction as the very difference between the RNA-seq. So, you know, whenever we take a piece of tissue, there's also a whole multitude of cells within this. So we get the bulk RNA input. And then when we get our gene expression back, we have this average gene expression from all the cells, and any cellular heterogeneity within that sample is masked, which obviously has very useful points about it as well. But with single cell, we put in each individual single cell, as the name suggests, and then we get this distinct expression profile per cell, and allows us much higher resolution of expression, identifying rare cell populations, and also some stuff that would be completely hidden because of the level of expression compared to the rest of it. If you have a cell population that's 5% of all of it, or even smaller than that, you would just completely lose this information in your RNA-seq. So for experimental background, with these genetically clonal lines, this system's extremely cool at INRAN. They have this whole selection of all these rainbow trout lines bred for different resistances to different aquaculture pathogens. So on the left, we have the VHSV, so this is a survival curve, and on the right, we have Flavobacterium. And this is just to kind of highlight this contrasted resistance. So the two fish lines that we chose to use were AP2, which is highly susceptible to viral infection, and B57, which is highly resistant, almost having 100% survival after 35 days, whereas the AP2 is completely wiped out after 10 days. And then the opposite side of things, we can see the B57 cell line, fish line is much more susceptible to bacterial infection, whereas AP2 is much, much more resistant. Experimental design for this part of the topic was to take two control fish from each line, and then we took the head kidneys from two of these, and then the spleen from also one matched fish. So we had the AP2 and B57. And then we also did the same, but with VHS. So two fish were infected with VHS from AP2, and then a further two for B57. These were left for 48 hours, and then we collect all these head kidney leukocytes using a fecal gradient to purify them. These get sent away for 10x sequencing, as explained in the title slide, and then we can proceed to downstream analysis where we do feature selection, identification of different cell types and subcell types, and have a look at their gene expression like that. So within this, we had two comparisons we aimed to investigate. What are the differences in cell types between unstimulated AP2 and B57? And what do these look like, gene expression-wise? And then following that, what are the changes in cell composition and expression between the control and VHS infected fish for each fish line? The single-cell data obviously produces a huge mass amount of data, so that kind of work is for another talk, and I'm just going to talk about the unstimulated AP2 and B57 fish. So following after all our downstream analysis, we arrive at a plot called a UMAP, which is a dimension reduction plot, and the idea being each single little point on the plot represents a cell. And the program tries to cluster these based on their expression similarity, clustering together or much closer together, or further apart based on the dissimilarity of the expression profile. So it still doesn't really tell us much at this stage, apart from we have 30-odd different clusters of cells identified by the program. So one of the first things we do is we annotate on the fish cell line, so the AP2 and the B57. What we did initially see is you see quite a good, fairly mixing, but some separation between the two fish lines and some of these kind of big globs or clusters of cells. But the main thing that really sticks out to us is this very high separation of blue and red, B57 and AP2 with these cells. But we need to start annotating actually what they are and describing what these different subcells are. So we use various different marker genes from canonical studies done in zebrafish and mammals to start identifying what they are. So MPX being a marker for granulocytes, TIMP2A being a marker for your monocyte populations, a hemoglobin gene is obviously red blood cells, even though we extract as many as possible with the leukocyte purification. Factor 13 being a marker for your thrombocytes or platelet cells. CD79 and IgM being part of your B cells. CD3 epsilon being T cells, and then CD8 being your cytotoxic T cells. So following on from all of this, there was obviously several different markers we use. We don't just rely on one output. We come to the dot plot, which explains the different markers we used, and then we annotate all the cells. So we managed to get 6 different lineages of cells, all annotated, from granulocytes to T cells, with one unannotated cluster at the moment, which has really proven a bit of a nightmare to actually identify what it is. There's this very general, generic gene expression here. What we did see, just a couple other points, granulocytes form the bulk of the cell populations in the head kidney, with the B cells being second, T cells. Some differences between the two lines, proportionally, with monocytes being higher in B57. There'd be more monocytes than red blood cells, but I guess this could also maybe be down to the extraction method between them all. Following on from that, with the big difference being in the granulocytes, this was obviously where we wanted to start. So what we can do is, because at that point we have the global gene expression, every different cell cluster or every cell lineage is compared against all the rest of them. But it doesn't really, it kind of masks a lot of the heterogeneity within these different subclusters. So what we do is we pull out the granulocytes and then reanalyze it and recluster it. And what we see is exactly the same situation that we've seen in the global UMAP encompassing them all. We initially started by trying to identify different subtypes of cells, such as your basophils and eosinophils, but we were completely unable to identify any of these subtypes within it. And what we did see instead was this kind of gradient of gene expression going from the top to the bottom, or the bottom to the top. So up at the top, what we've seen was more kind of immune gene expression, such as MMP13 and MPX, being much higher expressed. And this kind of dropped off in a gradient towards the bottom, whereas the bottom of these UMAPs, we see much more ribosomal proteins and transcriptional genes here in the cell cycle. And so we tentatively concluded at the moment we have a much more mature population of granulocytes towards the top of the UMAP, and maybe more immature transcriptionally active ones at the bottom. Between the two lines, we then had a look at some of the most differential genes here. And what we've seen was various different immune genes that were differentially expressed between the two of them. So CD106 being vascular cellular adhesion molecule, much higher in AP2, fibrinogen-like, higher in AP2. And then lastly, because here we have 2 paralogous genes of beta-2 microglobulin, which forms part of your MHC complex, with beta-2M2 being higher in AP2 compared to B57, and then the opposite being observed for B2M1, with it being very highly present in B57. Also, just of note, in B57, we've seen a couple of different interferon-stimulated genes, so IFA-M3, and another protein which is quite novel, AAP1, which is very highly expressed in B57, but almost completely absent in AP2 cells, which is kind of like a pore-forming protein. There's not actually much work being done on this. We then compare that gene expression, which was localized to the granulocytes, to the rest of it, to kind of, I guess in a way, show the power of the single cell, and also how some of these genes expressions can be very highly localized, and other ones are a bit more general. So with VCAM1, we see this very localized to your granulocytes, monocytes, and thrombocytes, with not very much going on in the rest of the lineages. Same can be said about EEP1, with granulocytes and monocytes taking the bulk of this expression, and it can more or less completely absent in the rest of the cells. But other ones, such as your B2M, we have differential expression levels, but is present in every single cell lineage that we had looked at. So just my take-home message and a bit of conclusions. We've seen, obviously, these clear cellular differences between the AP2 and B57 leukocytes, specifically in our granulocytes. And then more general for the kind of talk and the work we've done, molecular basis of disease response is vital for selective breeding of fish, for resistance, maybe other technologies such as your vaccine development, and then the use of these state-of-the-art technologies can only further increase our rate of understanding immune responses and improve prevention strategies. So I would like to thank all my co-authors, and special thanks to supervisors for put up with me for the last couple of years trying to finish this. Our aquarium teams at both INRAT and Aberdeen, and of course Horizon 2020 funding and AQUA-FAANG. And I'll take any questions.